Hello, everyone. We're happy to welcome you to the Human Cell Atlas Biological Network seminar. Today, we're featuring Kidney Bionetwork. We would like to remind you to join the Human Cell Atlas by visiting our webpage. We currently have 18 biological networks. You can become a member of as many as you would like. Please indicate your interest by visiting our website. If you're preparing publication that you would like to submit to be included as an HCA publication, please let us know. Visit again our website, and there are instructions on how to do that. This could help your paper gain more visibility. We would like to remind you of the upcoming HCA events. Please feel free to register and participate. We are excited to welcome you to the November HCA Asia meeting in Thailand. It's a hybrid meeting, so feel free to join virtually. The next HCA Biological Network seminar will be dedicated to data submission guidelines and ethical considerations. It's a virtual seminar that will happen on November 10th. The registration will open sometime next week. The HCA Development and Pediatrics meeting is coming up in the end of November in Paris. And HCA-wide town hall is scheduled for December. Also, next week we have a workshop for for single cell RNA-seq analysis for our African colleagues. It's the first workshop in the series. We will be announcing more as the time goes. So today's talks are from Menna Clatworthy, a 4D kidney cell atlas. Matthias Kretzler will present defining the cellular landscape of kidney disease across network and continents. And the panel discussion will be coordinated by Greka, who's coordinator of Kidney Biological Network. So I will gladly introduce Anna in a second. So, Anna, if you could come on stage, that would be great. Yeah. So, Anna is an associate professor of medicine at Harvard Medical School, a physician at the Brigham and Women's Hospital, and an institute member of the Broad Institute of MIT and Harvard. Anna leads a program aimed at dissecting fundamental mechanisms of disrupted cellular homeostasis in genetically defined kidney, metabolic and degenerative diseases with a special focus on membrane proteins. Anna, please take it away.
Helen, thank you so much for this kind introduction, and it's such a wonderful pleasure to be with everyone today. I see already we have 51 participants. How awesome. And really great to be with everyone today from wherever you are. So my job is simply to introduce you to two of my most amazing colleagues and fellow co-coordinators of the Kidney Bio Network, Menna Clatworthy and Matthias Kretzler. Just by way of very brief introduction, we formed this group several years ago now with the purpose of understanding the human kidney through time and across different ethnic backgrounds, the different aspects of the kidney, this organ that is so complex in its architecture. We often say only second to the brain in complexity in terms of its functions and architecture. And as you can see on this slide, we basically had multiple tools and resources that have been brought to bear in our network. And in addition, we've had a lot of different tissue sources from which we have been able to secure human kidneys for this analysis. And of course, as with all other networks, working on the computational tools to integrate these data, and of course also integrate the data from the kidney with all the other organs in this incredible human cell atlas that we're all working on building. And so many colleagues from around the world have been a part of this effort. And of course, we would like to invite anyone who wishes to join us. And so now, without further ado, I just wanted to introduce our first speaker. As Ellen mentioned, it's Menna Clatworthy. Menna is a professor of translational medicine at the University of Cambridge. She received her doctorate in medicine, also at the University of Cambridge, and she specializes in nephrology. Menna is a fellow of the Academy of Medical Sciences and also was elected to the EMBO in 2022. Her lab focuses on human tissue immunity and also houses a very valuable resource, the Cambridge Tissue Immunity Laboratory. Menna is really a remarkable physician scientist and someone I admire deeply. And it's really such a special pleasure to have you here today, Menna, and also an opportunity for me to thank you for always being a partner in this process. So thank you for being here today.
Well, thanks so much for your kind words, Anna. I really appreciate it, and also the opportunity to be able to share something of what we've been doing. So I'll just share my screen now, hopefully. Does that look okay?
Looks
That's
great.
perfect. Perfect.
Okay, great. So I've given my talk a slightly quirky title, and in fact I've put a question mark at the end of it, and you can see what you make of this title at the end, which is a 4D kidney cell atlas. So I'm actually a nephrologist, so a clinician scientist, as Anna said. So I'm deeply interested in the kidney, both from a basic science aspect and from the point of view of serving the patients that I look after, which are mainly in transplantation. And the kidney is an amazing organ. So anatomically, it's got an outer cortex, so shown here, which contains these filtering units, which are called glomeruli, and blood gets squeezed through loops of capillaries in these glomeruli to generate a filtrate, which ultimately forms urine. And this then flows on through a series of tubules that are lined by specialized epithelial cells into the medulla, which is a high salt tip part of the tissue. Water is reabsorbed using that high salt concentration, and ultimately a concentrated urine flows down into the pelvis and into the ureter, the tube that leads down into the bladder. And one of the things about the kidney, as well as containing these many different specialized cell types, both epithelial and endothelial and immune cell, as you'll see, immunological perturbations come to the kidney in very anatomically polarized ways. And I should say that my bias is actually in immunological diseases that affect the kidney and other organs. So what do I mean by that? Well, I mean that, for example, if you consider the main bacterial infection that comes up from the bladder and into the ureter and will affect the pelvis and inner bits of the kidney. But many immune stimuli that are circulating, the way that they come to the kidney is actually through the cortex and through the blood supply of the kidney. And many of them will initially affect these little, the filtering units, the glomeruli. And that can cause inflammation. And some classical examples of that are diseases that are mediated by autoantibodies, such as lupus nephritis. Diseases that are mediated by antibodies that react with bacterial products, such as post-infectious glomerulonephritis, or even in transplantation, antibody-mediated rejection. But all of them are characterized by antibodies that come in and cause inflammation, particularly in the glomeruli. So the way in which clinically we traditionally understand how this works is that we take a biopsy. So if somebody presents to us with their kidney not working properly, we take a biopsy of the kidney and we do light microscopy. And we simply describe the morphology of the cell types based on H&E staining, whether there are cells that are there that shouldn't be, like immune cells, like neutrophils are shown here, or whether there's evidence of immune cell activation as shown here, complement activation. But I would emphasize that the way we interpret this and the way we make diagnoses, a normal reference is critical. But overall, I would say, very critically that nephropathology clinically currently is very rudimentary, and we're still pretty 19th century in all honesty. But a way of making this better would be to do molecular profiling, to identify molecular pathotypes within the kidney. So this could be by bulk RNA sequencing, where we might say that there are sets of genes that are regulated in a disease state versus not. But of course, even better is single cell genomics, where you can identify in an unbiased way the different immune cell subsets that are present in the kidney and how those cell states might change in the context of the disease. But to make to understand what happens and what goes wrong in disease, you need to make a normal reference atlas, both of bulk transcriptomic normal reference atlas across different parts of the kidney, and ideally a single cell atlas. So with this in mind, we began as part of the Human Cell Atlas project to make our contribution to developing a normal human kidney cell atlas over lifespan. And this is what we initially did. We took 6 kidneys prenatal, 14 postnatal, and the source of these tissues were either tumor nephrectomies, the normal bit of it, or kidneys that have been donated for transplant but couldn't be used for a whole bunch of reasons. And we did single-cell RNA sequencing using the 10x genomics platform. And this generated about 27,000 cells from developing prenatal kidneys, and the different cell clusters are shown here. I don't need to go into that for the purpose of this talk. And then in the adult kidney, we could actually see that, from my perspective, very interestingly, there was a heterogeneous population of immune cells, including some we'd expected to see. So we expected to see mononuclear phagocytes, so macrophages and dendritic cells. They're known to be tissue resident. But we also found a bunch of adaptive immune cells like B cells and T cells, which maybe we weren't expecting to be there in normal kidney. And as well as that, we profiled the nephron epithelial cell types, endothelial cell types, and stromal cells. But one thing that I'll point out was perhaps slightly disappointing. So some cells that are really of interest, for example, the specialized epithelial cells that live in the glomeruli, these filtering units are called podocytes. And we had limited representation here, and even less representation of some of the other cells within the glomeruli, which I'll come back to. So what are we able to do with that sort of reference atlas? Well, the kind of studies we went on to do would be to think about some of the diseases I'm interested in, like antibody-mediated rejection, lupus nephritis, that are antibody-mediated. And we know that many of the pathogenic effects of IgG antibodies are mediated by binding to these cell surface Fc gamma receptors. And these are expressed in the kidney. We can now say, in an unbiased way, we take our human reference single cell atlas and say which cell types are expressing Fc gamma receptors. And what this honed down to was a lot of expression on macrophages, dendritic cells, and monocyte-derived MACs, as shown here. So we then took our data, our normal reference data, so the blue dots here, and integrated it with some published data on lupus nephritis biopsy, so an IgG-mediated disease. And you can see these were clinical biopsies. They didn't have so many macrophages, but if we integrate them with our normal reference, we can actually see these nice clusters of tissue resident and monocyte-derived MACs forming, and we can begin to ask questions about which cells are getting activated. So here, for example, we took a reference macrophage stimulation signature, and you can see in lupus that's enriched, so particularly in the classical monocyte-derived MACs. But that was kind of what we did with version 1 with our initial reference to try and understand something about antibody-mediated disease. But when we were thinking about the next phases, which is really where this network came in, what we really needed was more cells from more individuals. You know, we had 14 adults there and 40,000 cells. But also not just any cell or every cell in our sequencing efforts. You can see, so if I take you back to this UMAP, most of the cells that we've got here are these proximal tubular epithelial cells. And these are the most numerous cell type in the kidney, which is why we see loads of them, but we don't really want to be sequencing any more of these. You know, what we want actually are more immune cells, cells of interest in terms of responding to perturbations, and also some of these cells that are present within glomeruli. And if we zoom in and think about the glomeruli in more details, I mentioned the podocytes, right, these specialized epithelial cells, but there are also these supporting mesangial cells, and as well as that, glomerular capillary endothelial cells that are specialized capillaries. So really we wanted to try and get more coverage of these cells, you know, to provide a proper reference for glomerulonephritis, for these diseases that cause inflammation in the kidney, including antibody-mediated inflammation. So as we began to put our kidney seed network together, unfortunately the pandemic struck. And for us, that made things very difficult because first of all, one of our sources of kidneys, which was kidneys that had been retrieved for transplant, really became very limited because transplantation, because of the ITU bed crisis, really shut down more or less. And secondly, the way that we were processing our kidney samples was that we were processing everything fresh. So taking chunks of kidney, making single cell suspensions and loading them fresh onto the 10X machine. And that required lots of people. So usually there were 4 or 5 people in the lab working on a kidney when we got one in. And that just wasn't possible in the pandemic because we were only allowed 1 person in the lab at a time. And so we adapted, that pressure really made us adapt what we did. And we tried a bunch of ways of trying to get it so that 1 person could take samples, could freeze, and then we could thaw them and multiplex them at a later date. So do everything in a much more controlled way. And we tried this in 2 ways. The first was to generate single cell suspensions, freeze them and then thaw and then load. And then secondly, to actually take chunks of kidney, freeze, then make single cell suspensions and load. And it was the latter that gave us the best results. And then the other problem was enriching for these cell types of interest. So I mentioned the glomeruli, right, these filtering units, and we want to get more of these cells. So what we did was turn to actually something that was done historically with physiologists studying glomeruli. And that was to use the size of the glomeruli and the fact that you could use sequential sieves. And although cells would get through 100 micron sieve, the glomeruli actually stay on top. And so you're able to get isolated glomeruli and then make single cell suspensions and do single cell RNA-seq. And the other thing we did was the tubular interstitial fraction. What we've been doing is to sort out the proximal tubular cells because we don't want to sequence any more of those. So we used a marker CD13 and used a flow cytometer to sort those out. And then we also used a trick where we could load multiple different individuals because we had these frozen samples. So we could defrost, say, 4 or 5 samples at one time, mix different genotypes within lanes and using a leave one out strategy. That would then enable us to demultiplex what was in each lane without prior knowledge of their genotype, so without a reference genotype, so making that a lot easier. I'm just showing that using this, we were able to identify the different genotypes that we put into the experiment. So ultimately, then our version 2 was much more pleasing in terms of the heterogeneity we were able to get in our reference data set. And we were also able to give an anatomically annotated data set. So that is, we know which cells came from our glomerular isolation. So we're shown in orange in here, and which were from the tubular interstitium that was proximal tubular cell depleted. And so you can see really nicely here a whole bunch more of mononuclear phagocytes, many more than we previously got. Again, some more podocytes than previously, still only a small number of mesangial cells, but quite complex in terms of the endothelial cells, and the glomerular endothelial cells in particular we're able to capture. So this is really pleasing. And now we could, because we know where these cells came from, right, either the glomerular extraction or the tubular interstitial extraction, we can then see the location, the anatomical location of each of these, so a spatially resolved kidney cell atlas, we might call it. So there are a bunch of cells we would expect that we would find in the glomerular fraction, for example, in the non-immune cells, the mesangial cells, right, or the podocytes. But then when we plot the immune cells, this is really interesting. So what we see is that in the glomerular fraction, there are a whole bunch of monocytes and macrophages, as well as NK cells and NKT cells, whereas the lymphocytes overall seem to be in the tubular interstitial fraction. And we can then validate this using spatial transcriptomics. So we haven't done loads of this, but this is one of our early pieces of pilot data, where we've got a piece of kidney. This bit didn't work so well, but we were able to annotate known anatomical regions. So for example, the glomeruli, the proximal tubular cells, fibrotic regions, which are shown in these red regions here. And then we were able to parse out the different cell types that we'd found in our higher resolution atlas into those different structures. So again, for example, podocytes are found here within the glomerular spots. But you can see, for example, in the fibrotic spots, there are a whole bunch of immune cells, including things we perhaps wouldn't expect, like B cells and plasma cells. Now, going back to my long-standing interest of antibody-mediated kidney diseases, so this has been good in that it's enabled us to get a reference of more glomerular cell types, which is what we need for a glomerulonephritis. But one thing it doesn't allow us to do is to understand how the glomeruli got to that point in lupus nephritis, right? So what we want to know is the earliest changes that might occur in kidney immune cells and non-immune cells in response to a perturbation like circulating autoantibodies. And we can do this in mice very easily. We can inject intravenously some immune complexes, as we did here in this experiment. And then when we harvest the kidney, we can see an upregulation of different pro-inflammatory cytokines. And we can do flow cytometry and understand which cells might be contributing those. But how do we achieve this in the human? And we realized that actually something that we do clinically and in transplantation, we do this all the time. So we do this to assess kidneys. We put them on an ex vivo perfusion rig. And this means that kidneys get perfused with oxygenated, warmed, packed red blood cells through the artery, so the normal blood supply, and actually some of them on the rig will start producing urine. So this gives an opportunity to potentially administer immune complexes and set up a dynamic model. So that's why I'm calling it potentially a 4D response cell atlas. So in the interest of time, because probably I'm slightly going over, I will skip this, but we've done this now in 3 kidney pairs, and we're able to use one kidney as a control, doesn't get immune complexes, but it's of course genetically identical to its pair, and the other gets immune complexes. And we use fluorescence immune complexes, so we can see by flow cytometry, so these are A647 labeled, that there are cells that take up immune complexes in both cortex and medulla. And we can use flow to identify some of the cell types in a biased way. So here we're looking at macrophage markers, and we see that there are macrophages that take up immune complexes. And we can also see there are NK cells and B cells that to a lesser extent do that. And we can confirm those immune complexes are internalized by doing confocal imaging. So cyan are the immune complexes, 206 is a macrophage marker as a CD14, and you can see some of these on the inside. But then we also took a number of samples for RNA sequencing, and what we did was the same trick we'd done to make the reference atlas. So we isolated glomeruli and negatively sorted the proximal tubular cells. And here's some of the glomeruli that we got. So you can see the beautiful loops and some of the immune complex labeled cells within those loops. And this is the data that we got. So again, we can divide it up into glomerular versus tubular interstitium, but also this time from immune complex stimulated kidneys versus control. So we're beginning to generate this immune complex response cell atlas. And we can use neighborhood analysis to look at which neighborhoods increase with immune complex stimulation. So you can see there are changes here among the glomerular endothelial cells, among monocytes and among NK cells. And we can probe this in detail. This just shows the monocyte macrophage response. It doesn't matter in detail, but just gives an idea of how we can do this. We can look at the cues that some of these cell types might be producing in terms of cytokines and chemokines that might signal to other immune and non-immune cells, as shown here. And we can use cell-cell interaction analysis. So for example, we use cell phone DB here to think about how monocytes as the sender of the immune complex stimulus might send specific cues, for example, chemokine cues to receiving cells. So overall, we can build up a complex signaling network of how human kidney resident cells, both immune and non-immune, respond to a challenge. So overall, I would say that, as part of our network, COVID significantly affected what we were able to do initially, but we adapted. And actually overall that improved our capacity to multiplex samples. We've also developed this glomerular enrichment strategy, which provides us with compartmentally resolved atlas data, which I think is really helpful and really interesting from the immune cell point of view, as I showed you. And then we're now exploring trying to make a 4D atlas. So that is, how do cells respond? What's the delta? So with that, I'll finish and thank those that were involved. Here's my lab and the people who were specifically involved in just taking these kidneys and processing them, particularly during the pandemic, which could be quite lonely. So that was Kevin, Naton, and Georgie in particular. And Ben Stewart, who's a PhD student who's just finishing my lab, really has driven a lot of the analysis that I've shown you here and has been really amazing. We're really reliant on the organ donors and their families to give us organs, as well as patients who are undergoing tumor nephrectomies. Of course, these are the institutes that house my lab, and also I thank our funders, and will be happy to be involved in discussions at the end. Thank you.
Menna, thank you. This was a remarkable tour de force, I would say. And just a reminder for those in the audience, now we're up to 70 participants. Please put your questions in the Q&A and I'll be happy to read them out. I do have a question to start us off with. And I guess, you know, one of the obvious things to ask about, first of all, I totally buy into the 4D. I like it, you know, the whole paradigm. I think it works, and I think it makes perfect sense and something that we need to continue to do. But one other aspect of the atlasing that I think many are wondering about is, you know, what about beyond the transcriptomics? Maybe something to comment on, and maybe it will come back again in other discussions. But especially when I think about immune cells, you know, it seems that a lot of what they do, their business is to present things on their surface and interact with other cells, right? And so thinking about the protein level. So what are your thoughts on integrating other methodologies and building upon this 4D map that we're creating?
Yeah, so I think that's right, Anna. I mean, I guess that pragmatically, the way that we've all come to this is that measuring transcripts in high dimension is just way easier than measuring proteins in high dimension. And so that's been the thing that technically people have been able to embrace, both initially bulk and now single cell. So whereas high dimensional protein imaging, where, you know, protein analysis is way more difficult. If you look at something like mass cytometry, right, which has been used maybe in an analogous way to this, to say single cell RNA-seq, where you generate a mush or a suspension with no knowledge. And so there, of course, you can profile thousands of proteins, whereas say for bulk RNA-seq, it would be 20,000+ transcripts for that sort of thing. For mass spec, it's about maybe 4,000 or 5,000 proteins. But the problem with those sorts of analyses, they're dominated by housekeeping proteins, so many of which are not that interesting. Okay. And then the other thing is, of course, the spatial element, you know, which cells are actually producing those proteins. So then I think the most powerful things to come to this will be methods to spatially identify proteins. And I still see it as you use the transcriptomic data as a discovery platform to, you know, to come up with some hypotheses or some pathways that you think are important, proteins that you want to validate, and then come up with something that's lower dimensional. I don't just mean 4, I mean, you know, in the region, you know, you can do imaging mass cytometry, or you can do IBEX, the strip staining. So I mean in the region of 30 to 50 proteins, spatially resolved, that are maybe there to try and validate specific questions or hypotheses. Maybe that's how I would see it.
That makes perfect sense to me, and I think that's a great way to think about it, and you know, a way to move forward, I think, for many. There are some questions in the chat, so let me try to tackle some of them. A quick one: do you have experience working with post-mortem kidney?
So short answer is no, I haven't personally. The samples we have started to try and use are clinical samples that have been taken, so like a kidney biopsy that we do on the ward here, and then it's formalin fixed, paraffin embedded. We've started to try and use those. It can be challenging to get adequate quality RNA out, but there are potentially now, so for example, 10x has a Visium platform that potentially allows you to do some spatial transcriptomics. I think post-mortem, the samples that would work from talking to colleagues have to be relatively rapid post-mortem. And at least I can say in the UK that the standard hospital post-mortems are not rapid. They happen several days after. Mm-hmm. So I think the standard post-mortem doesn't work. So it would have to be some sort of fast-tracked post-mortem sampling. But I think it's a bigger, the debate is about what's the best tissue source for a normal atlas or even a disease atlas. And I would say that every tissue has its advantages, or every tissue source has its advantages and disadvantages. If you're taking biopsies from patients, the reason they're getting a biopsy is because they have a problem, right? So even if it's adjacent normal, there's a question of how normal. And because they're alive, the piece is often tiny. So that's the disadvantage there. For the transplant, discarded transplants, obviously they've undergone a period of cold ischemia when they've been in an ice bucket, so they have a disadvantage. For the post-mortem people, well, they are dead, which is, the transcriptional. So I think we have to try and gain the truth from all of those, and all of them have their strengths and weaknesses, I would say.
Yeah, that makes perfect sense. And I think we are going to hear next from Matthias, who also has this incredible resource of being able to biopsy normal individuals who are willing to contribute to research. And that's quite rare, but I think he will share with us some of the learnings from that as well. So I think, I agree that tissue there's pluses and minuses to every approach. Yeah. There is a question about multiplexing, and it is, can you expand on the advantages of multiplexing? Presumably the cell number per sample reduces proportionately. And so if pre-selection from the tissue isn't done, rarer phenotypes might be missed. How much do you gain by increasing the multiplexing? I guess
so I think that's absolutely right. There's a sweet spot. The advantage of the multiplexing is one, just a practical, pragmatic thing. So, if you're having to run 2-3 lanes of 10x for every person, right, it becomes non-feasible just from an expense point of view. So there are practical advantages. There are also technical or analytical advantages from multiplexing individuals into the same lane. So for example, the ambient RNA is then identical for those multiple individuals, and that kind of batch effect, you're not getting that. But you're absolutely right. You do want a minimum number of cells from each of the subjects contributing to that to be able to gain sufficient insights. But I think for us in the kidney, I mean, there are people, and I think even in the chat I can see. Yeah. there are people like, for example, one of my colleagues in the UK, Donald Fraser, who's very interested in proximal tubular cells, right? So whereas I'm trying to get rid of them because I don't want to sequence any more of them, then I think it depends on your question, but you may well need enrichment strategy, so you've got enough of the cell type of interest represented. But yeah, you can't multiplex endly, because one, computationally, deconvolving the genotypes without reference becomes difficult if there's a tiny number of cells. So that's true. So we've tended to do about 4 to 5 individuals per lane.
Yeah, that does make sense. And it was actually Donald who was asking the question. I'm trying to catch up with everyone here. Malta is asking about tissue dissociation protocols and other sample handling decisions and how those kind of affect the various cell types that we detect. I think that's a great question.
Yeah, so definitely, I mentioned that because of COVID, we were forced into adopting a freezing strategy, and I would say, freezing does, when you freeze thaw, you kill some cells. So definitely already you introduce some bias there. Potentially the cells, so in the kidney, I didn't mention this, those proximal tubular cells that I mentioned are pesky and numerous. They are very metabolically active, so potentially those are, and those are the ones that are most at risk of ischemic damage in vivo in our patients. So potentially those are the ones that are most at risk during the freeze-thaw procedure, and also during the whole. For us, we get at least half of our kidneys, as these are kidneys that are donated for transplant but can't be used. So at least half of those have a period of cold ischemia, and again, the proximal tubular cells, we know that can portend delayed graft function if we go on to transplant. So it's the proximal tubular cells that get down. So yeah, so I think so how if the sample processing involves a period of freezing and thawing, then inevitably that will get rid of some of the proximal tubular cells. In general, we find that for single cell using, and we've principally used the 10x platform, that this, I think, favors sequencing of immune cells, and T cells in particular, I think just seem to survive the whole thing very well. So which is great for us because we're interested in immune cells, but may not be so great for other people. But, and again, I showed you the data, so, and I should say, when we isolate those glomeruli, and then we use quite an aggressive enzyme mixture to then digest to get into the single cell suspensions, you know, we're still not getting great coverage of mesangial cells. We're getting more than we did, but you know, I think that there's something that we're doing that we're not capturing as many of those as we should. So you know, when you present a talk like this, you give the headlines, but the number of experiments and the hours that go into trying to optimize what, you know, really tedious things like what's the mixture of enzyme you use, or how long do you do it at room temperature or cold, you know, yeah. Right. So all of that is incredibly important, yeah.
Absolutely. And hopefully that also answers Tomer Kaliski's question about kind of the same idea of like, what's the right protocol for frozen tissue? So you covered some of that, but yeah. I also should point out that many of our labs are posting on Protocols.io, and a lot of these protocols are out there for people to look at. But of course, all of us are happy to answer any questions, I guess.
Yeah. So we've got our initial protocol is on protocols.io. We've actually now done some really very recently done some multiome from you know, nuclei in a multiome, and we'll put that protocol out. But I think a number of groups have done that. So yeah.
That's fantastic. So I think maybe this would be an opportune time to move on to our next speaker, and then I think there's an opportunity for more discussions at the end. So let's do that. Thank you again, Menna. This was just absolutely phenomenal. And now we're in for another treat. We have Matthias Kretzler, the other co-coordinator of this effort in the kidney. Matthias is the Warner Lambert Park Davis Professor of Internal Medicine and Nephrology and Computational Medicine and Bioinformatics at the University of Michigan. His group has really been at the forefront from very early days in the use of multiomics in the kidney. The overarching goal of his research is to really define chronic organ dysfunction in mechanistic terms and use this knowledge for targeted therapies. And to reach this goal, really over many years, he has assembled incredible databases and groups of scientists who work together. I would say that you know, Matthias kind of showed us the power of team science before the rest of us really understood just how important that would be. As a result, Matthias leads the Kidney Precision Medicine Project, Central Hub, and this Kidney Atlas mapping project. He's the leader of a U54 in Nephrotic Syndrome Research Network, the Neptune Network, which has been incredibly important for our understanding of kidney disease. He's the PI of several additional networks, also focused on understanding kidney disease from a team science perspective, CureGN, for example, where he's the director of the Applied Systems Biology Core. And he's also the PI of a large systems biology effort, NIH-funded effort to understand diabetic microvascular complications. So he has really been at the forefront and a leader in all these areas of using multi-omics to understand the kidney. And of course, I have long admired Matthias for this incredible ability to bring all of us together and work together to understand kidney and develop therapeutic strategies, and really thank you for always leading the way in that regard. So without further ado, Matthias, the floor is yours.
Anna, Menna, I hope you can hear me well. Hey, thanks so much for that very kind introduction. Now, it's an incredibly exciting time to do team science, and really kudos to the Human Cell Atlas and the Jane Zuckerberg Foundation to really bring the international community together to make an impact for, you know, people suffering from so many problems, which I think with the emerging cutting edge technologies you have seen so beautifully introduced by Menna, you know, we can and we do have a real impact for real people today. As Menna already introduced to you, we feel, and there is a conflict of interest statement down here that a renal anatomist and nephrologist is speaking here. We feel that the kidney complexity in structure and function is only second to the brain, And this is a great organ system to learn how complex three-dimensional epithelial structures interact with vasculature and immune cells. And as Menna also told you, we actually can gain access to that tissue in many different ways, so that we have an opportunity to use the cutting-edge biomedical researchers material from real humans with real diseases to answer real questions with impact. And that has been the motivation for us really over the last couple of decades to really come towards a molecular definition of our diseases. And as Menna mentioned, up to now, we used a pretty descriptive disease definition of pattern recognition in histology with some basic clinical features to tell our patients what they are suffering from. And this is changing thanks to what Anna, Menna, and many others are doing in the field right now, really by the day. And the approaches here is, as Menna has pointed out to you, that we can take advantage of the fact that we take these kidney biopsy, these little pieces out of the kidney, and we can study them, and we can study them in histology. And this has been revolutionized as well, as we have now digital histopathology available, so that the images from a kidney biopsy can be generated, stored, and globally shared. And as I will see you right now, this allows collaboration seamlessly across continents. Another feature is that we get biofluids, blood, as for any other organ systems, but the urine is particularly powerful, as we can find in the urine what's happening upstairs in the kidney with proteins, with metabolites, and even with cells who are shed from the kidney and can be recovered in urine samples. For example, the group from Anand Arasi and Celine Bertier from our team who is on the call, some beautiful studies showing how macrophages change their genotype with single cell sequencing from blood throughout the lupus kidneys and into lupus urine samples. And then this fourth armamentarium we have added to our study research portfolio is the ability to, from people where we have that tissue level definition of disease and the time sequence of disease from the non-invasive biofluids, we can capture cells. And Anna has been spearheading a lot of our efforts in using these cell-based systems in what we call kidneys on a chip, organoids, who are highly differentiated 3D structures who allow you to capture the cellular phenotypes present. And as Anna has shown from MOOC1 in a beautiful study from 2019, if you want to see the power behind these systems. And in many of our cohorts, we have this package now available to generate, starting with clinically phenotypes, and in many instances, several thousand parameters captured over time, structure, and then obviously the genetic makeup and the multiple data elements coming together. And with this, we can follow in our cohorts over time on a cohort level what's happening to our patients, identify those who do poorly under standard of care, and with the tools and toys Menna has introduced to you, we can understand what pathways are active in people who are not responding to the treatment options we can offer to them today. Very exciting piece, which I will not have time today to introduce to you, but we are doing in our rare disease network. We are now doing it on an individual patient level, that we take that knowledge out of our knowledge network, we identify representative key disease drivers present in a patient's kidney, we capture them out of blood or mainly urine samples actually, and then tell a patient from the emerging therapeutic clinical trials, and this is the fourth exciting piece in our field, that we see finally now trials coming back to kidney and kidney disease research, which patient would benefit best from which potential emerging therapy and should be recruited in a specific trial. There's a personal story behind that as well, which I would like to share to you, particularly to the young investigators, that it's really worthwhile to stick to your guns, to really stay with your technology and your expertise. In 1994, actually during my fellowship in Michigan at that time, we established protocols how to get tissue pieces out of the kidney and to use microdissection and quantitative RT-PCR. It was really labor intense, but we were able to scale that down with patch clamp biology to actually harvest individual cells. Here's the filter cells, the podocytes Menna has introduced to you, and do individual molecule, individual cell quantitative measurements. And then obviously took advantage as different technologies came around, both in the data generation with the microarrays and then the data deconvolution. This is with Olga Troyan-Skolyova, who is also leading the data integration efforts with Rachel Silphon in her team in the Human Cell Atlas Seed Network. And then obviously we were able to utilize both the expertise and most importantly the samples we have generated over the last 20 years with the emerging technologies you have been introduced today already. And the framework which we have established over the last couple of decades now allows us to study these molecular disease definitions which we are seeing emerging in different networks across the continent, with the initial efforts initiated with EU funding in Europe and then the rare disease networks in North America. And very excitingly, and we have been very fortunate to work with an incredible group of dedicated researchers in sub-Saharan Africa and the H3CKD Africa network, who have brought these technologies, which we purposefully defined to be robust in the field, literally into the jungle in sub-Saharan Africa, and has generated now beautiful cross-sectional and prospective cohorts of tissue available for analysis, including for analysis, maybe the Human Cell Atlas might benefit from as well, to capture obviously the incredible genetic diversity there. And then based on these experiences, we now have an effort in place funded by the NIDDK, and I see Eric is on the call today, who leads the effort from the NIH Institute here to bring these studies from these rare diseases to the common diseases in acute kidney injury and CKD. One key feature which Menna and Anna have alluded to as well is really, if we have so many different technologies, is there a way to anchor these technologies against the reference tissue? And Michael Spitzer here at the University of Michigan in nephrology, together with urology and pathology, has developed a very effective accessing program where we generate from unaffected parts of tumor nephrectomies with really minimal tissue perturbation large pieces of tissue, which we then process in multiple different processing protocols using nano printed devices to actually generate elements which resemble pieces similar to biopsies, which we then can share widely. And this has been one of the resources for the Human Cell Atlas Kidney Seed Network, which we were able to procure even while the pandemic slowed our and other procurement efforts down to somewhat. Joshua Levy from the Standard Working Group in Human Cell Atlas is on the call as well, and we managed to establish the data sharing of the same kidney pieces with a standardized working group for this network, who is establishing different technologies, and again have the advantage that kidney is driving the science forward here and can be mapped together. And we are using the same reference material also for the Kidney Precision Medicine project, which I will show to you in a moment what we are doing there, so that we can combine then different technologies on what will be very small sample sizes for these technology development programs, but they will be able hopefully to fill in some gaps where an individual technology is not as informative as we hope it to be. What I would like to do over the next 10 minutes or so, I would like to introduce to you to the strategy we have developed and now implemented in the Kidney Precision Medicine project. This might be of help for many other research networks who are focused on an organ system, how to set up a network and how to leverage a lot of the expertise and protocols who are in the field. The goal of KPMP is along the line Menna defined, that we want to capture disease mechanisms, disease subgroups, to define new treatments, but we are taking advantage of the fact that we can obtain kidney biopsies. And we feel, and we, I think, have documented over the last 15 years that this can be done safely with a minimal risk to study participants, so that we can offer this as a research protocol procedure for patient volunteers to help to drive science forward. And that really is a concept I really want to bring to the Human Cell Atlas as well, that we should really engage our volunteers who are providing tissue for our studies in our efforts. And KPMP really has developed a very robust framework where our patient participants contribute to all aspects of our science. And it's always very grounding to hear from patients suffering from the diseases why we are here and what we are doing, particularly to the non-clinician scientists in the teams. And with these patient scientists, we really have developed an approach now where we use from routine kidney biopsies or in KPNP from research kidney biopsies, the protocol which Menna and I and Anna do for clinical care delivery now in a research setting to obtain small tissue pieces. They are 12 millimeter in length and roughly a millimeter in diameter. And we process them in a highly standardized manner. These protocols are available in Protocol.io. I will show you the link in a moment. And then we bring these samples to the different technologies from the same source to generate information. Obviously, this will be a small sample size, but with the different technologies, we can use it complementarily. One key feature is, I think, still relevant in 2022, the structure and how we can structure using our conventional histological processing, the presence of structural alterations in the kidney tissue. It also allows you to do rigorous QC on the incoming material to ensure that tissue integrity is present. The pathology group has developed a set of manual descriptors, which are semi-quantitatively quickly scored and are available then for correlative analysis with clinical and most important, the molecular-based system. And then obviously we see a plethora of AI-based automatic image analysis platforms going live, which is very exciting and can allow us to capture specific segments in the kidney, can allow then to define distinct subfeatures in these segmented areas who are reflective of structural changes, and if automatically and high throughput assessed, will be available for correlative studies with the molecular assessment. As I mentioned, the key feature in KPNP is to capture a maximum of technologies, which really also maximum of quality control, so that the user of the information, you have access to all of the metadata relevant for the data generation, so that if you define any specific features in the data set of interest to you, you always can bring it back and identify definitions of participant, specimen, and data generation as experts. And with this, we have developed now a comprehensive set of technologies which are sequentially activated to be used against tissue donation from our study participants to allow us to build a comprehensive picture of the multi-scalar information present in the kidney tissue in the KPNP diseased kidney tissue atlas. And an atlas for whom and for what. So as a software development team here in my group in Michigan, and I see Nikki and Becky are on the call and available for discussions later on, establish comprehensive user personas with semi-structured interviews to really learning what our different users for the atlas really want to get out of it. And you can see we do have our study participants as a main user, so they can see their kidneys and what we're learning from their kidneys in a patient participant portal. And then obviously the different users, many of you on the call here will be expert research data miners, but a main user group also is the non-expert bioinformatician we want to serve around the world going forward. And for that, we build a structured atlas design with heterogeneous data inputs, and then very importantly, heterogeneous data outputs with tools which we develop specifically for the kidney community in KPNP, but many other tools like Cell by Gene or the CCF Explorer, the Common Coordinate Framework from HubMap, which are built across organ systems, but kidney can populate one element in these organ systems, and with the data sets we derive, we can inform some of the cost-cutting efforts there. I invite you to have a look how we set up the system, both to engage participants, the community at large, both clinicians and scientists, and then also the various data mining tools. Critically for resources, as Menna already mentioned as well, is that we share our protocols early and often. So what I have shown you, and I will continue to show you, is all available in Protocol.io under KPNP, including very detailed step-by-step hands-on procedures, how to use frozen, how to use formalin fixed, how to use snap frozen material for the different technologies. If you have any questions, please give us feedback. These protocols are there to be used. Same for the metadata standards, which are purposefully harmonized to whatever extent possible with HubMap and Human Cell Atlas, so that the data is informative when you find a change in a specific parameter, so that you can track that back to the clinical processing or analytical standards involved in here. And then we have also reagents, particularly for the spatially resolved protein data. As Menna mentioned, this is a key feature where I think we will learn a lot, that we have a list of validated antibodies who work on FFPE and frozen sections. We have built a series of data sharing resources, data atlas repository for you, the power users, to download the raw data. That repository is openly accessible, and only a few data files who would allow patient identifications are behind a simple DUA gate, so that you attest to us that you will not try to re-identify individuals. Otherwise, all the data shared in the atlas are fully and effectively accessible. We have an explorer tool for the clinician scientists, cell biologists without specific bioinformatic expertise, to quickly get down to the key pieces of information you need. And then together with the Vitesse team from Niels Gelenberg, we have a spatial viewer now live, where you can detect the molecular information and the structural features we have. The interface is kidney specific, kidney focused, it's dynamic. If you can select your cell types and then dive into the various data sets to give you information around their cell type, give you summarized data you can visualize and subset for the distinct disease subtypes. The spatial viewer, we have the histology scans in there, which I think are very helpful if you have a background in pathology to link the features what you are seeing. We have 3D cytometry imaging coming from the Indiana University team live, and we have a rapidly growing set of spatial transcriptomics and code examples available as well. If you want to dive into the transcriptomic and proteomic spatial signatures captured from people with healthy reference material, acute kidney injury or chronic kidney disease. The key feature is to link that information together. In KPMP, we obviously did a lot of work in our what's called ontology mapping together with the human phenotype effort, GoodMap, RBK, NIDDK efforts, and the NIH HubMap efforts to map complex clinical information, molecular and structural information together, so that we can project these different elements on towards that. This requires a robust data processing pipeline. I'll give you the reference here where we have described that with significant detail and each of the quality steps involved, so that the data integration and output in the end actually is meaningful and allows you to do that crosswalk between the different technologies. Here in the Science Advances paper is a beautiful example how for those pesky abundant cells, the proximal tubular cells, but also critical for many pathophysiological and physiological function of the kidneys, we now can use transcriptional, proteomic, and metabolomic spatial data to learn and teach us how different cell population in the tubules do their energy generation and transport work in the kidney. We can then map that along the different nephron segments. The glomerulus would be here, who produces 100 liters of urine, and you obviously only have one coming out in the end. So there's a lot of work happening along the nephron, which we now can map in that spatial context. We can tell what energy for these processes is coming from anaerobic or aerobic glycolysis, and also predefine areas of maximal vulnerability where cells doing a lot of work but have limited access to oxygen. And those are exactly the cells we are seeing now in the biopsies from people with acute kidney injury, a lot of changes being happening. And that, I think, is a probably very helpful reference also to show how data from Human Cell Atlas, HubMap, and KPMP from various sample sources and various technology sources can be combined for a multimodular map of the neighborhood and niches of the Human Cell Atlas. This is work done led by Blue Lake and Raji Menon. I think both of them are on the call and available for the discussions in a moment. Where the team across these three networks mapped roughly 500,000 cells from 80 people, define their three-dimensional arrangement, define neighborhoods, and then use that information to define not only cell types, but cell subtypes, how they are activated. And this is now visible in the Atlas Explorer, where you can go in and you can look where these different cell types live. how they change between health, acute and chronic kidney injury, how they change their phenotype, and what molecules go up and down. Interestingly, the spatial data also allowed us to go one step further to not only tell us which cells are expressing which molecule and what potential receptor ligand interactions are taking place between activated epithelial cells and inflammatory cells, or the other way around. But now with the spatial data, we actually can validate which of these interactions are physically possible because these cells live in the same neighborhood. And as we have defined these different activated and damaged neighborhoods, we then were able to take that signature out of the KPMP single cell dissolved data and project it with deconvolution strategies developed by the team into our rare disease network, where we have 15 years of microdissected bulk RNA sequencing data available. And now we can see which of these activated cells states are actually associated with poor long-term outcome. And very much to the surprise of the community, we see that yes, the proximal tubular cells, if they are damaged, that's not good for you. But the thick ascending limb, those are the later stages in the nephron who have less energy access, appear to be the cell stage, which is strongly predicting outcome. And in the atlas, if you want to find out what does this nephron segment do, you can click on the anatomical definition of that segment. You get the molecules are selectively expressed in that segment versus others. So for example, number 3 on that list is an epidermal growth factor, EGF. It's a secreted cytokine, secreted growth factor, which we actually have shown in a lot of studies can be actually measured in the urine as well, and correlates with urinary measurements to the tissue level. And this was a study with Wen Chun Jun in our biomarker core, actually initiated over 15 years ago, where we now have measured over 20,000 people who have an activation biomarker, the EGF, obtained and associated with outcome out of what now is a spatially defined context. And based on the KPMP data, the AKI research team in KPMP already did a study and could show that not only for CKD, for chronic, but also for acute kidney injury, that molecule predicts not only who gets the injury before cardiac surgery measurement in the urine, but that these urine measurements even predict who will have lasting kidney damage in place, showing a first snippet how this information can help us to manage our people better. I would like to wrap up in really hopefully having made a critical claim for it is worthwhile to build the virtual villages where, you know, the Human Cell Atlas really has been such a tremendous example for the scientific community at large. And to bring different people from different backgrounds with different expertise together so that we can define those key features we have to attack in disease mechanism towards targeted treatment so that we can help our people with the disease, what we are doing our work for, to keep them as active as possible in this setting. And this is really my main thank you to all of our patient participants, our patient volunteers over the last 2 decades who has made that work possible. And also obviously to the team science group here in Michigan who throughout the pandemic kept us pushing forward to bring new insights to people with kidney disease. And with this, I thank you for your attention, and I'm very happy to take any questions.
Wow, Matthias, I knew you were amazing, but you're Captain America with the kidneys in the middle. That's pretty awesome. I love that. The t-shirt in the picture. This was really another tour de force. Again, as a reminder to people to put questions in the Q&A as we begin our conversations. There is actually a question in the chat, and maybe inspired by that, expand a little bit further. You mentioned how building this atlas can now be used with other databases, such as the Neptune cohort, for example, to deconvolve various signatures and understand mechanisms within those samples collected over many years. So the question in the chat is about whether there's children with Bardet-Biedl syndrome who are in that cohort, and whether there have been any insights from this analysis. But I would expand that to say, can you comment further on whether you think that this foundational map will allow us to understand fundamental mechanisms either in rare or common diseases of the kidney, and maybe talk a little more about the therapeutic strategies that may be inspired by that? Thank you.
I think this is really, again, it's a fantastic opportunity for team science, and both for the Human Cell Atlas and for KPMP, Rachel Seelphone and Olga Troyanskoyova from the Ziegler Institute for Systems Biology in Princeton are working closely in these networks to bring these different data sources together. Because yes, we don't have biopsies from kids with Bardet-Biedl syndrome, but we know their genetic makeup, and Olga and Rachel have developed beautiful tools how to use comprehensive whole genome sequencing to predict actually your single cell activation states, and to therefore determine not only if your variance you are seeing is significant or not, but also in which functional context that variance is most likely working. And they are critically dependent on data sources like the Human Cell Atlas and KPMP to feed their algorithms and to make them more specific. And the other piece, and Anna can speak to that because she is really at the bleeding edge of that research, is that if you don't know exactly what's going on, there is a way to have ex vivo organ models now with the inducible pluripotent stem cells you can grow out of a simple blood sample. And Anna has done, and Anna, you might want to speak to the MOOC1 story, because I think this is a beautiful example how using these systems combined, one can develop a targetable strategy, which then can bring treatments to people with rare and even ultra rare diseases. The final comment is, these data are all publicly available, and I feel strongly that they have to, so that researchers both in academia and industry can bring their domain expertise to these large knowledge bases, where each and every one of us only scratches a small segment of the information that is present in there. And I really, again, want to congratulate Human Cell and Payne Zuckerberg to really bringing that concept of open science, open community to the forefront, and thereby bringing everybody along. Because if key efforts like this one share openly and freely, then that makes it a lot easier and actually expected from everybody around the globe.
Yeah, that's fantastic, Matthias, and I agree. This concept of lifting all boats and making sure that we can all work together to make discoveries. And it is true that I think some of these foundational resources, like you mentioned, also single cell atlases of organoids, together with the human kidney, and with mouse as well, that has been well characterized, can all be tools that can help us in our discoveries of pathways and targets that can ultimately be targeted for therapies. I want to open up to any other points that either Menna or you, Matthias, would like to make as we are approaching. Oh, we have one more question. Oh, the question is about Bardet-Biedl again, and whether there would be iPSCs for those. And I guess I will leave that to you to take offline. I wanted to ask, sorry, go ahead. Yeah.
Then I think the point is to obviously identify research lab who have domain expertise in that area, which we don't. We are working primarily on nephrotic syndrome diseases in the rare disease network, but there are plenty of researchers in that space available, and reaching out to them and finding out how and where you can help with your translational research interest, and conversely what insights, including potential therapeutic trials they have active and live on.
That's right. Yeah, I think that's kind of the point of having more open science, so that people can access this information, and there are groups that are working on this. I wanted to open it up to any final comments from either of you. There was some discussion earlier on that we didn't fully address about other types of data sets in addition to transcriptomics. We didn't talk so much about ATAC-seq, for example, so that there was one question that I think if there's time we could possibly address, and how it can be integrated with other data sets that are being generated. I don't know if either of you will have comments on that, but also just open it up to any other final comments that you may have.
Yeah, I mean, I think I would see, Matthias probably has some thoughts as well, but I would see that it will be useful for us to generate ATAC-seq data. So I said we'd had a crack at generating some multi-ohm data across the whole of the depth of kidney. And I think that maybe speaks a bit to what we were talking about, thinking about proteins and I suppose the functional significance. And I guess that's what the chromatin landscape in some ways gives you is, some idea about how the genome is poised for transcription and maybe that can give you insights, potential functional hints. So I think, but then it comes to one of the challenges which I think we all face, right, as we're trying to address questions, which is, what's normal, I suppose, how does normal change across lifespan? How is it different between sexes? How is it different between different ethnic backgrounds? And how does that change with disease? A challenge is as we get more data is just how we deal with it, right? The scale of data now, and as soon as we start putting a spatial element into it, that's even storing that data is hard, or analyzing that data. So I think that is perhaps where some of the challenges will be. How do we develop systems to practically handle what will be massive data sets, even on an individual patient?
Yeah, this is where I think obviously Jane Zuckerberg is uniquely poised to develop cutting-edge solutions to this problem, and I think we have a great opportunity to work also with the software engineers here from CCI at various places to really push towards an open science, open community who also makes these data accessible. And really bringing algorithm to data, I think, is a unique advantage, which also really democratizes research significantly. And it's exciting, with our Sub-Saharan Africa network in HCA. Now the investigators there, they access the data. They have access to cell phone, even if they don't have access to running water, but they can access these informations, and we can have clinical pathological conferences in that setting. And they can drive a lot of the key questions forward, which only they understand from the context of the disease as it presents it to them in the patient populations they are serving. What is very important is that this is obviously very sensitive information, and that we have very careful communications with participants, patients, how and where we garner and provide access to data, and we really facilitate effective use, but obviously prevent or at least minimize misuse of information. And that obviously will be a constant push and pull as we see the power of the insight we can gain from our studies, exponentially increase.
Very good. And I think that may be an appropriate place for us to end, in the sense that, we have done a lot of good work and we can be happy with what we have accomplished, but also there's always more to do on many different fronts, increasing the complexity of our samples and our data, understanding how to handle that better, understanding the ethical implications of the work that we do, making sure that we are true to our commitment to capturing the diversity of the human population in our efforts in creating this atlas, not only for the kidney, but of course, far beyond that. So maybe that's an appropriate place to end, with the motivation to continue to work on all of these open fronts with team science, as we have been doing until now. And I think our bio network has been, productive, and, hopefully we can continue to look forward to that as we move forward. So from my perspective, I think this comes to a natural closure for our session. I think Ellen is keeping us organized by thanking everyone for coming today, which I also, of course, would like to thank everyone, and then also reminding us of some of the upcoming opportunities. So unless there's any further comments from you, Ellen, I'm very happy to Again, thank everyone for participating today, and especially Menna and Matthias for taking the time to take us through their incredible work. And thank all of you for your time and participation. So thank you again very much.
Thanks, everyone. Thank you. Goodbye.